Right, good morning, everyone, and welcome here. We're coming from far, and welcome first time at our campus as well. Also, the people at home, thank you for joining this seminar by Claus Taylor, who I found out today that he is Belgian-British, but working in Denmark. He's coming over from the Center for Protein Research, affiliated to the Novo Nordisk Foundation. Previously, he did a postdoc in the lab of Peter Lyman in Switzerland, in Texas, but he was doing a structural work there. And before that postdoc, he was a postdoc in Spain, in the CSIC in Madrid, also doing structural work with Professor Fernando Tornero. So he's a structural biologist, and he will tell us more today about structural work he has done, injection machineries, and so on. So I expect a lot of fantastic, nice pictures and so on. So people at home, you can put your questions in the chat box, so we can address them at the end of the seminar, and also for the people here in the room. I propose we take all the questions in the end. Then I pass the floor to you. And
thanks a lot, Yves, for the very nice introduction. And I hope I won't disappoint too much, because I will talk a bit mainly about bacterial motility, but there's a very interesting link with bacteriophages, but you'll have to wait till the end to find out. Oh, running into difficulties.
Ja,
en dan is het erachter moeten halen.
Okay. So yeah, so as we all know, biological membranes are both the barriers as well as the interfaces of our cells. And nature has evolved different protein machineries to deal with this compartmentalization. So some of these can be encoded. Some of these systems are encoded by the cell themselves to transport molecules across the membrane, but also some are made by attackers, for example, bacteriophages, to transport, for example, genomes into. What we want to understand in my research group is how these molecular machines can transfer molecules across the membrane barrier, how these systems are regulated, and also in the longer term, how they might be adapted for biotechnological or biomedical purposes. And to do that, we mainly use one technique, one described electron microscopy, which most of you have heard of course. And its recent advances, or relatively recent advances in detection technology, that now allow us to visualize at molecular detail some of these machinery. So the scientific question that I will talk about today is how bacteria move. And why is it important? Of course, because movement allows bacteria to explore different niches. And that's important, for example, in the process of disease, when bacteria infects, they want to have, or they can only colonize specific niches because they have certain receptors, or in ecology, for example, oceans are full of bacteria that have to traverse large distances. So motility is very important. And about half of all bacteria are actually moto. So this plays a very big role in biology. Most of the bacteria that move use a large system known as the bacterial flagella for their movement. And they can swim. Here you see, for example, E. coli swimming around happily. And as you can see, most of the bacteria swim in a straight line. And so the way that they do that is that they have several flagella in the case of E. coli, and they beat all together in one direction, which is counterclockwise. And all these flagella will make a large bundle that works together to rotate, and the bacterium will swim straight. But as you can see sometimes, for example here, these bacteria start tumbling around and then change direction. And that's how they change the direction. So by tumbling around and then swimming off in a random different direction. How do they do that? What happens on the molecular level is that there is chemotactic signaling that will result in phosphorylation of one signaling molecule known as KY that will then somehow break up the flagellar bundle because some of the flagella will now turn clockwise instead of counterclockwise. The flagellar bundle breaks up and the bacterium just starts stumbling around. And then it will go in a random direction. But because this process is more likely to happen under unfavorable circumstances, on average, the bacterium will swim towards what it favors and away from what it doesn't like. So that's how it works. But of course, we don't know how it works at the moment. So what does the flagellum look like? It looks a bit like this. So here we see the schematic of a bacterium with its flagellum. So the flagellum is a very large filament, can be over 1 micron long, consisting of one protein, flagellin, that is a polymer, and it is connected through a flexible hook, shown here, to the molecular motor, and that's what powers the rotation. So it's a rotary motor. The rotary motor consists mainly of a rotary part here, also known as the basal body, and it is surrounded by different things that have been thought to power this rotation, known as the stator units. So stator units consist of two proteins, MotA and MotB, and they form up some kind of ion channel. So in E. coli or Salmonella, these proteins, MotA and MotB, form a proton channel, and they use the proton motive force that is generated in basically all bacteria across the inner membrane. So in most bacteria, you would have a proton motive force or an ion motive force across this membrane. So ions can flow through the ion channel, and this somehow powers rotation of this flagella. In some other organisms, we have, for example, in the sea, there's a lot of sodium. So there's sodium motive force in most bacteria, and we have sodium powered stator units. Before, this complex has been proposed to have a stoichiometry 4:2, and I will come back why this is important. And just to remember, there's one very important residue, which is aspartate on the MotB supplement. Usually, the channel is in its inactive state when it's not incorporated in the motor, and that's important because otherwise it would disrupt the ion motive force, and then the bacteria will die. So you don't want that; it should only be activated. So it's thought that the channel is plugged and becomes unplugged when it's activated. MotA, which is shown here in colors, MotB is black and white. So MotA is interacting with the rotor through this interaction with flag G. And it was thought initially that binding of ions to MotA and MotB introduces some conformational change that then powers rotation of the rotor. But we did not understand how that happened. So what we wanted to find out is how the stator unit can actually power the rotation of the motor, both in its natural counterclockwise direction, so the bacterium can swim straight, but also in the opposite direction, so that the bacterium can change the direction. And this was the work from several people in the lab, mainly from Monica Sandivari, but also from Aris Heide, in collaboration with Mark Erhard and our Prof, who recently actually passed away. And so to tackle this question, we use structural biology. And first step we often need to do is to get a lot of protein because we want to study the protein. So to do that, we purify large amounts of our membrane protein by cloning, expressing, and purifying it using affinity chromatography and then gel filtration. And we work at high concentration, but less high than, for example, crystallography. So it's around 1 mg/ml. And then we look at it using Cryo-EM. So we vitrify our sample by rapidly plunge freezing in liquid ethane cooled by liquid nitrogen, which allows for very rapid freezing so that we don't have crystalline ice, but we have this vitreous ice, so glass-like ice, and we can visualize it using Cryo-EM. So that's on here. So we apply our sample and rapidly frozen. Then we will collect our data at high resolution microscope, and using data processing, we can come up with a map of the electrostatic potential of our molecule, into which we can build an atomic model for our structure. that can hopefully tell us something about the biology, which is of course what we want to find out. So this complex, MotA, MotB, it is quite tricky to work with because it's a membrane protein complex, and these proteins are more difficult and they need to be isolated from the membrane. And that can be challenging. And so to help that process, we had to screen a lot of different organisms to find the optimal protein complex to work on. So we screened in total 8 different stator units, and out of these, only 3 actually behaved relatively well when isolated from the membrane. That is shown here. So 3 of these protein complexes were then amenable to single particle Cryo-EM, and they all eluted more or less the same molecular weight, which was around 300 to 400 kilodalton, including the third amino. So here you see how pure they are. So this is quite good for membrane proteins. Sometimes we see these higher molecular weight bands that correspond to multimers of membrane proteins, because often for membrane proteins they don't completely disassemble when incubated. And so just to show you what we get after all the processing, that I won't go into detail, but I can of course happily explain more, is that we get, for one of these, for Campylobacter jejuni status unit, we get a very high resolution structure to around 3 angstrom, and we get several states, and most of what I will present is about this state, organism. But also for Schwannella onadensis, as well as this MotAB stator of bacterial, which is a sodium stator, we get relatively high resolution structures where we at least can see the secondary structural organization of this motor complex. So this is just the pipeline of the whole processing. As I said, I won't go into a lot of detail, but basically we start from our cryo-electron micrographs, and then we select the particles from our homogeneous preparation, and then we start classifying particles to get rid. So to keep the best particles and to classify the structural heterogeneity. So the sample was very homogeneous. So unlike with a lot of other projects, we didn't have to classify so much, and we could actually get a very nice resolution structure. So let's get into that structure. So here you see the organization, the secondary structure organization of the MotAB complex. As I said, it consists of 2 proteins, MotA, which is in the inner membrane containing 4 transmembrane helices, as well as MotB, that contains a single helix. MotB also contains a peptidoglycan binding motif, which is important because that's how it anchors the motor complex to the peptidoglycan. The structure of the peptidoglycan binding motif was known and determined before, but all the rest was unknown. Here. Here you see the structure of MotAB in its plugged inactive state. So just by purifying the protein and determining its cryo-EM structure. Important, or the most obvious first fact is that we observe that the channel actually consists of 5 copies of MotA surrounding 2 copies of MotB. And as I said before, this was unknown because it was thought that there would be 4 copies of MotA and 2 copies of MotB. But this creates some asymmetry, which is very important for its molecular function, and we'll come back to that. Are we sure about this 5:2 stoichiometry? Yes, because we do observe it in all 3 of our stator units. So even these are lower resolution structures, we can still see the secondary structural organization, and we can clearly observe the 5 copies of MotA surrounding 2 copies of MotB. So apart from the overall organization of the complex, when we now look at the conservation of the different parts of this, we see that at the bottom of the MotA stator unit, we see a very conserved patch of residues shown here. So they are colored just switch up, but the red is very conserved part. So, and this is at the bottom of the study unit here. So when we then correlate this with some mutations that have been described in the literature, we propose that this is the interaction site with the big rotor of the flagellar motor and with this phage helix that has previously been found to have some mutations that are complemented by other mutations inside this helix. And we see this charge interactions here, and all other people have found before that you can switch the charges and still remain or have active flagella. And we also investigate further using biological experiments, namely swimming assays. So for the people that don't know what this is, because it's very specific, so basically you have a very dilute agar, so it's like around 3%, and then you put some bacteria on it, and if your bacteria are able to swim, then they will swim a bit, and they will start growing, swim, grow, and they form these halos. That's why you see, like here, halo of bacteria, and if they are not able to swim, you just get like a dense. So that's how people do it. As I said, the channel has to be inactive because otherwise bacteria will die. And the way that they keep it inactive is with these two things known as the plugs. So this is plug 1 and plug 2 because you have these two MotAB helices that lie flat, and when they get inactivated, they bind the peptidoglycan and the motor becomes active. But we don't understand yet how. That will talk about it. That's also shown here. So we have introns stopping and then activation by binding to peptidoglycan and by dimerization of this peptidoglycan binding domain to enable peptidoglycan binding. So we were, of course, interested to find out how the complex is really working. So we thought if we believe this plug, then maybe we can activate it, as has also been shown by other people before. So what we did was we made a deletion of the plug region by removing these residues, 41 to 60, which corresponds roughly to this, and has been shown in E. coli to activate the complex for ions. So we looked at the structure, of course, using cryo-electron microscopy, and to our surprise, we saw very little differences, except for two things. One was this active site residue, this aspartate 22, that we think is binding the ion, and that's important. The second one, however, was unexpected and kind of expected. The second one was unexpected, and it's a hydrophobic residue, phenylalanine in this case, that lies at the top of the channel, and that we think is gating the access of ions to flow through the channel. And I will show more later. So that's great, and that shows the non-protonated state. But as I said before, the hypothesis in the field was that protonation of this complex, the activated complex, would result in conformational change that can then like drive rotational flagella. So to try to visualize that, we tried to make a protonation mimic of this complex. So to do that, we mutated aspartates, in this case 22 for our Campylobacter, to asparagine, and tried to have a look if there was any difference. So to our very big surprise, there was actually no difference, except for one residue, this aspartate 22, that we saw that instead of pointing towards the periplasm, it now points towards the cytoplasm. But that was the only major difference that's also shown here from the side. So we did not observe any large conformational change in MotAB, asking us, or leading to us asking what could actually be responsible for the activation of the rotation. And we had to come up with a different way of trying to understand how the flagellar motor is powered. So as I mentioned before, the first one was this large conformational change, where MotAB changes between two conformations, like a kind of flipper thing. Or, and this we think now is the most likely, that there's a rotational model where MotA would rotate around MotB, And actually, MotAB is a miniature rotary motor turning the larger rotary motor. And there is a lot of things that talk in favor of this. For example, when we look at the interface, the symmetry of the large rotor. So this is just representing the symmetry of the flagellar motor. So imagine the flagellar motor sticking, the flagellum sticking out here. We see that between successive of the flagella segments, we get distances that would correspond very well. We see an ion consumption that is quite supportive of this model. And this allowed us to, unfortunately, we cannot really say if the rotation step would be 36 degrees or 72 degrees because of not enough accuracy in the determination of ions consumed. But we favor the system where it rotates 36-degree steps so that each of the MotB molecules alternately engages with the proton and rotates the motor. And I will show in a movie how that works. To come up with this movie, we basically assume that each MotB molecule in different states of the process takes up in the next step the conformation of this next one. So they change roles all the time. And to visualize that, we can rotate this one by 180 so that this one in the next step. Yeah, it's a bit, it gets a lot easier when I try to explain this with the result, but I'm of course be happy to explain if anybody wants to. So the way that we think it works is that we have a MotA, that is this large membrane protein surrounding 2 copies of MotB, and that MotA is actually very, very hydrophobic, but it needs to rotate around MotB, which is very charged because it has 2 aspartates. So we think that rotation can only happen if this whole thing is neutralized. And in our model, each aspartate is able to bind sodium, but at any point there's at least one aspartate interacting with an ion and engaging with MotA. So we call this the engaged one because it's engaged to MotA. So what we think then happens is that this one is a high energy state and it wants to rotate, but it cannot because of the charge. But when the proton motive force now allows the when the proton motive force now allows that this aspartate get neutralized by taking up this proton from the periplasm, is now neutralized, and now the conformational change can take place, which is the rotation of MotA in a clockwise rotation of 36 degrees. Now allowing this aspartate to release the proton that it already had to the periplasm. Then there's conformational change that allows it to take up a new proton. And again, as this is a high energy state, and now it's neutralized, there can be again a 36-degree rotation step. And so this process keeps on being alternated, and you have a net clockwise rotation of the MotA around MotB. And we can also look at this just from the structural part. So here you see MotAB. We looked at it through the inner membrane. Now we're looking from the periplasm. 2 copies of MotB surrounded by 5 copies of MotA. And this is just the net rotation without showing anything, but just explaining the different states. But then after this, we will zoom in and we're going to visualize these 2 conserved aspartate residues that are part of the MotB. So zoom in. Now you see the 2 aspartates, and so we will put a blue halo around exactly here, around the aspartate that is engaged and like catching on to the MotA and doing the power stroke. So now this one catches on, it does the power stroke, and so this one gets neutralized, allows the power stroke of the blue one, and so on. And so that's how we think that motility works. So now, of course, we're like also collaborating with other people to try to visualize this. So that's great, because now we have a model for the unidirectional powering of motility. So we can swim in one direction because we have something that rotates clockwise, and it could push our motor. But it does not allow us to tell how it can switch. So let's have a look. So we have our clockwise motor, and as I said before, most bacteria or E. coli swims counterclockwise when in the normal conditions. So how can this happen? It could happen if our stator units are interacting with our big rotor or the flagellum, and the clockwise rotation of the stator units with these peptides, these flag peptides, interacting with these charges, as I explained, pushing by clockwise rotation and engaging here, it would push the rotor to the counterclockwise direction. So this would keep the normal straight swimming. If all the flagella do that, they will make a bundle and they will swim straight. So what we propose now that happens is that upon chemotactic signaling, this key Y molecule will get phosphorylated and it will bind to the rotor, and the rotor changes its conformation. It will widen up, and it's accompanied by 180-degree flipping of the flag peptide, as has been shown also through crystallography. So if you look here at the charges, we have red and blue, but basically this peptide flips like this. No, like this. So that now we have the blue on the left and the red on the right, and we have an expansion of the rotor. And now the clockwise rotation of the same clockwise rotation of MotA around MotB now pushes the rotor also clockwise. So if some of the flagella are going to do that, the flagellar bundle will break up, the bacterium starts tumbling and will swim in a different direction. So that's how we propose that the bacteria can change its swimming direction. So this was our work, but at the same time, Susan Lee and colleagues came up with the structure of also the MotAB channel, but the inactivated state only, but still they got the same model. And also there was some tomography studies by Young Yu and colleagues that I present here that are at lower resolution, because using tomography you cannot obtain the same high resolution, but you can visualize systems inside the cell, which is also one advantage. So I'll just go start again. So here you see the flagellar motor with this is the visualization with the stator units spinning clockwise and the motor spinning counterclockwise. And this is what they observed is a conformation change that they modeled here by after binding of QY, that there is this expansion of the rotor. Here you see that. So instead of engaging to the inside, you engage to the outside, and the same rotation of the rotor. You can do the same. So all of this evidence combined, so our studies and this study, as well as the crystallographic studies I mentioned, kind of point all to the same direction, where we have a very elegant way of powering rotation in two different directions just by engaging to the side of the same molecular complexes. So this was our work that we published before, but then there are still some open questions that we do not understand. So one thing is that now we looked into these stator units and also Susan Lee of the proton-dependent motors, but we don't understand yet the sodium-dependent motors. So one thing is, of course, we think they are quite working the same way. However, we are interested to review the ion selectivity mechanism. However, I think it's especially important because it would allow us to visualize the coupling between this ion binding and the movement, which we cannot observe at the moment because we're looking at protons, which are very small, and we cannot solve them using Cryo-EM, at least not until we get to very high resolution. But we can with sodium. However, we tried very hard, and also Susan Lee tried. But for some reason, we were stuck to around 6 angstrom for these sodium-dependent motors that I also presented before. However, then Highlight in the lab started working more and more on this, and at some point he was able to actually get a really nice resolution structure of MotAB, unfortunately only in inactivated state with the two plugs lying on top. However, for the first time, we got a nice resolution here, also showing that our model of the plugging was correct, where we have this trans plugging, because there was actually some difference with the structure exactly in this region. So we show that. One thing that is quite striking is that there's actually quite a lot of asymmetry here, which we did not observe in the other structure, and we're still thinking if there might be some biological role about this asymmetry. But one thing we could at least get some insight into is how ion specificity might be gained, because for the first time we could observe ions because now they are sodium. So we see one sodium in this side and one sodium in the other. So this would be in the engaged subunit that I said before. So there's a very close interaction of this sodium. So for the first time we prove this. We see this actually, we visualize what we hypothesized before on the protein structure. And we see an additional sodium side, with the caveat of it being in the inactive state. Nevertheless, we see a nice coordination of our sodium as expected, also with 5 binding partners. And we see that both POM-A and POM-B are important for determining specificity for sodium. The other side is much weaker and it's only POM-B interacting. When we do molecular dynamics, we see that actually this sodium is very stable, the other ones can go. Which is also kind of supporting our mechanism of the release of the sodium. So here we visualize the structural basis for the specificity. So here we see a sodium-dependent stator unit, and here a proton-dependent stator unit, and you can see when we color here by hydrophobicity that there's actually a alanine here, and here it's isoleucine that we find. So the alanine in the vibrio. And here we see a threonine, so that here we have 2 oxidants that are needed to bind the sodium ion that we do not observe here in the proton motor. So and that's how we obtain this. And we also show that using swimming assays to prove it also biological. So one thing that we did observe is this asymmetry that I talked about. We do not understand it yet, but we think it might have actually a big role because we see this distance between the red and the blue parts of so the different charges with positive and negative charges. So we think it might be important for catching this flagella peptide, and that by regulating this distance, it can catch the flagella peptide for transport. But this can have 2 roles, and we didn't make a big deal out of it because we don't know which one. So one thing could be that by catching the peptide, this would activate the channel somehow. That's one option. But the other could also be that it would regulate the catching during the rotation, so that you only, or that it would help to catch while this is rotating, that only one of them is actually binding, and that it would open up when it's time to release the flagella peptide. So it's one of the 2, but we don't know which one. But it's subject for further study. So as I mentioned before, we would really like to get an active structure of the sodium stator unit, because then we would be able to visualize sodium, how sodium drives rotation of the stator units, and now we only have the inactivated state. For POM-A we were not able to do that, but now Highlight also got some structures of another protein, MotPS, and for this one he was able to get the activated structures using these plug regions. They are a bit lower resolution, but still should allow us to visualize some ions, and we're currently working on that to improve, and hopefully we can come up then with even more better mechanism. Oh, and now the moment you all waited for. Some bacteriophage things, but I think it's also quite nice. So, as you all know, bacteriophages have evolved various resistance mechanisms, or bacteria have evolved different resistance mechanisms against bacteriophages. So, the best known of those is CRISPR-Cas, but there is others, of course, and more are being discovered all the time. So, different abortive infection mechanisms, there are signaling systems depending on the cyclic oligonucleotides that have been discovered, and so on. So why did we get interested in? Partly because I'm interested also in bacteriophages, but also because we found, or highlighted, was browsing the literature and found that there's one system, one bacteriophage defense system, that actually encodes two proteins inside itself that are known as Zorya and Zorb, and that are homologous to MotA and MotB from here you see the flagellar model. So initially, this was described in Doran et al. From Rotem Sorik's group, so in 2018, where they discovered several novel systems. And these proteins here, Zorya is homologous to MotA, and Zorb is homologous to MotA. And they discovered type 1 and type 2. Type 1 has 2 extra proteins, type 1 has 1. And later on, Ghent et al. Describes one other system where you also have 2 additional proteins. So we were, of course, interested about this system, and we want to find out more how it works. Doran et al. Already did some experiments. They just show that basically everything is important, and all mutants basically block protection against phage. So Heide got interested into this, and being structural biologist, he tried to purify protein and look at it using electron microscopy. So initially, he purified this, and he looked at it, and he thought it looked horrible because you have this long. It's maybe difficult to see, but you have these long lines. It doesn't look very homogeneous because you have these little blocks which look a bit like MotB, but then you have these long lines, streaks lying all over the grid. So I think he did 2 preps, and he didn't even look at the sample using high resolution cryo-electron microscopy because he thought the protein had unfolded or something like that. However, at some point he visualized the sample, and he actually got a very high resolution, one of our highest resolution cryo-EM structure, showing, well, maybe showing that it's always good to look at your data, but also showing this very nice structure where we have something that looks exactly or very much like MotAB, this channel. For the first time also, you see we got into black and binding domain that I always said it's there, but you could not see it because it was disordered. But now it looks like it's closer to the channel, and that's probably why you see it. And finally, you see quite a long tail emanating from here, which is part of Zorya. And there's 1 alpha helix for each of the Zorya. So it's 5 alpha helices extended. And it's quite long, I don't know, maybe 50 angstrom or something, I would guess. But actually, when we look at our protein sequence, so this is 300 amino acids, maybe built for MotA, but we see that actually we have around 800 amino acids. So there is still quite a lot not built. And then when we visualize our sample also using Cryo-EM, we see that there's actually, the tail is much longer. And also we can build it, model it using AlphaFold, and it would correspond to around 800 angstrom. So we think there's an 800 angstrom long tail inside the cytoplasm of the cell. So that's for Zorya. For the other, so we decided to focus on this type 1 system. So we now have the structure of Zorya, but we then also want to look at Zorc and Zord additional proteins to find out how they might activate phage defense. So the first one was Zord, which we were able to purify and get a structure of, which you see here. Here you see our model. So what we observed was that by building the model and comparing it to different structure models in the database, that it's quite similar to, so that we find a similarity to chromatin remodelers, so that it might bind to DNA inside the cavity. And also we find helicase activity predicted and a nuclease activity predicted. So we already proved now that indeed it is a nuclease and that it's able to digest, for example, T cells. For the other proteins Zorya, we were also able to purify this quite nicely and get some kind of structure, but it's much more challenging and not as high resolution. However, the AlphaFold model fits very well. But one thing that we were able to predict, and we are currently testing also using in vivo or in bacterial experiments, is that maybe part of this Zorya protein is able to interact here at the bottom of the tail. It might be important for signal transduction. And so that leads us to propose some kind of mechanisms. And there's two ways, and we don't know which one is correct, but this is the first mechanism that has also been kind of suggested by Dora, where we have a ZORAB inside the membrane, just like the MotAB. The phage is infecting, and somehow the DNA is recognized by ZORD, and this makes a complex with the nucleic acid. This activates Zorya that we show is being able to bind to ZORAB, and this would activate ZORAB, allowing the flow of ions. Like I said, this would kill the bacterial cell, so that basically the cell dies right after recognition, and you would have a abortive infection. However, this is just one model, and the other model would be that actually somehow ZORD and ZORC, ZORAB is the sensor, and ZORC and ZORD are the effectors of the system. And so we're currently also collaborating with Simon Jackson to find out more about the mechanism. So we're comparing like effect on different phages, we're checking the escape mechanisms and so on. But yeah, I think it's very interesting. And whatever we find out, I'm sure it will be very nice mechanism. So with that, I would like to summarize the flagellar work that I presented, and show you how, and I showed you how the activated stator unit powers the rotating from bacterial motor in its natural direction and other one. Usually, the stator unit is not incorporated; it will be in the plugged state, and it will not conduct ions. Upon incorporation, this activates stator unit to engage to the peptide ligand, and it can now conduct ions. Flow of ions will turn the stator unit in clockwise direction, and the motor turns counterclockwise. Upon chemotactic signaling, QI will get phosphorylated, bind to the rotor. The rotor can now bind to the outside of the stator unit, and the same clockwise rotation of the stator unit now turns the flagellar motor also clockwise. So the flagellar bundle breaks up, and the bacteria change direction. So with that, I would like to thank everybody in the lab, our collaborators and the funding agencies, and thank you for your attention.
Thank you very much for this very interesting talk. Fascinating to get insight in how this small molecular motors work and with an hijack by infection to protect against phage. We have time for questions for the people here in the room. Also, the people at home, they can use the chat box. Questions from Niklas.
Have you maybe also seen resistance mechanism just for phages that work through the flagella? So there are maybe some phages that use the flagella to bind, and
Yes.
then maybe that there are some stages so that it cannot do that anymore. Or
Yeah. We haven't really looked into that, but I know that there is some phages and then sometimes you have mutations that make the flagellar motor stop turning, and then they cannot bind the phage anymore. So it can be an advantage to shut off motility to survive to protect against the
And so the mechanism how the rotor turns, I'm just wondering how conserved it is between the different bacterial species. Is it pretty conserved, or is there variation?
So we think it's universally conserved. So it is different, for example, than in archaea, where you have this archaeolum. So it's not really conserved with that, but all the flagellar motors, we think the rotation mechanism is the same. There are some differences depending on where you have, for example, on where you have the motor. So some bacteria only have flagella, and they might only be able to swim like this, for example.
Okay.
So there are different ways, but the way that the motor is powered, it's.
Thank you.
Maybe. Maybe from an engineering perspective, I was wondering the size of the peptidoglycan binding domains. What's the size, and is it really also specific for each organism, or it's binding more like conserved regions in the peptidoglycan?
Yes. No, I mean, I think they are quite related, but sometimes. Yeah. Sorry. Yeah. Well, maybe this. Yeah. So they are quite related, but sometimes there are more interactions between the stator units and the rest of the flagellar motor, so that it anchors better. So, and that's often seen in motors where you need higher torques, like in more viscous media, that you need to anchor. Yeah.
And then with the MotA, it's or MotB, it's only binding when it's as a dimer, or do they also bind separately, but then maybe in a lower affinity? Or
so all I can remember now by heart is that there's crystal structure as a dimer of it bound to some fragments of sugar.
And with the Zorya system, it's always bound, or it's also activated.
This we don't know, and it's very like what we kind of want to find out, because what we do observe right now is that so we don't have these plugs, it comes out straight, but we think it's negative because there's some charge interactions keeping it connected. So we think there must be, it's not activated, and there must be something happening. So either it's this binding that activates it, or something on the bottom stopping its interaction. But we don't understand. So, but this we think also it's in active state.
So in fact, what I find very amazing is that you have this very big macromolecular complex with lots of subunits. Is there anything known of how they would be assembled? Would you think about co-translational assembly, or in another way also, if, for example, you do the expression of these A and B subunits, do you have to do co-expression to have them fold properly? Because maybe one protein needs the other to fold properly. So
yes,
yeah.
So for most flagellar mode, like Nicola, I think there's like 4 something different operons doing the work. I think there's for making the basal body, for example. For some systems, you find them also only in one, but it's more rare. What we found for MotAB, for MotAB, where you only the 2 proteins, is we always co-express them, and we see this is important. Also, where we have the tag, they're always found together in operon A and B. So very likely, it's indeed important for the correct assembly, and that makes sense because B is inside of A, so it makes sense that you at least probably would need some B around for A to form.
Yeah, I was very interested by the asymmetry of this one complex.
Yeah.
But do you see it only on the potassium?
Sodium.
On the sodium channels? You only see it there?
Yes, yes. So we do, and
So-
we don't know. We don't know if that's.
Could it just be that maybe because the sodium is bigger, that it needs some space, so that creates some space while turning?
Yeah, it could be. We need to have a look. It is bigger, but it's also not dramatically bigger. So,
Yeah.
like we don't have enough structures to say it's because it's a sodium. It's just by chance. And it's, or maybe even possibly some kind of artifact. But it's definitely something, yeah, we want to.
It's cool.
I was wondering, so you're visualizing these proteins, but then normally they're incorporated in the membrane, and you're visualizing them without the membrane, I guess. Could there be a difference between those two states, and maybe also with the asymmetry, that when it's incorporated in the membrane, it has a slightly different structure?
Yes, that's a very good question, and it's a concern for all membrane proteins. So what we did for MotAB is we isolated with detergent. So usually you have lipids still around, but there's always a risk for delipidation. What we did for POMAB, so the sodium one, most of the structures we have the structure we have it in detergent, but we at the same time also incorporate the nanodiscs. So we extract with detergents, but we put them again in lipid nanodiscs where you have these protein rings surrounding lipids and then the protein inside. And we see basically the same structure.
I have two more questions, Nicholas. First, about the motility system. You explained us how it's activated, but is it on and off? Also, are there different speeds or gears? Can it accelerate? Is it always completely on, or are there intermediate stages?
Yeah, it's a good question, and it depends a lot on the type of bacterium. But for example, what I think on the small rotor level, so on MotAB, this is probably just dependent on time. So, but what the bacterium does and to regulate is two things. So one is it has some extra proteins, like brake proteins, like that can decouple rotation and block things like a real brake. So that's one. The other is by incorporating more or less of the stator units. So especially, for example, E. coli or Salmonella, you can have only one stator unit powering the whole thing, and then if you need more, incorporate more, transcribe more.
So it does mechanisms to do a sprint or a marathon.
For example.
The second question was about the phage defense system. I understand the post model, but I do not understand why this still should be that long. Because the mechanism that you explained, there's no need for one tail.
No, exactly. So we're also wondering, there is some Zorya systems with shorter tails. There is some hypotheses, maybe one hypothesis could be that it's more beneficial to be more in the center of the cell, or maybe this, if the phage is injecting, maybe something that's detected is more here, because after the
Intuitively, I would say the closer to the cell membrane that this can happen, the better, because the quicker you can hide.
Yes.
But then this protein, see, it has to bind in particular the region of the structure or around the region. I think it's wrong, and then bind.
Yes, so that's another thing that it might, if whatever. So we think this is our C, or we saw that this is our C probably binds here, but maybe this could bind something else. I don't know, maybe. And then there might be multiple things that combine. So we see some repetition here. So it's consistent there with something like that. But we need to.
Could it also be, instead of going to the center, that it's more shifted towards the side, cover a larger area?
Yeah.
Like this, or?
Yes. So it's like it can, maybe it's inflexible and can just like detect.
Yeah, that is
actually
actually
around.
sensing like a sensor or something.
Yeah. I guess it will keep us busy.
There might be a question on the chat.
There's a question from the chat from Ariana. She says, "Great talk, thank you. related to the previous question, which I could say. Do you know if general or dedicated chaperones are needed for the assembly of the complex?
Which complex?
Well,
Yeah.
she doesn't say.
But probably the flagellar motor.
I guess, yeah.
Yeah. We don't know, so the only thing I can say is that we overexpressed MotA and MotB as one operon in BL21 like cells. I guess it was C43. So whatever chaperone is needed at least to get this state is just standard equal chaperones.
All right. Then no further questions in the chat box or in the room. Then I thank you once again, Nicholas, to give us this fascinating.
Oh, there's still one question from Bjorn. Bjorn says, "Cool work. Thank you for the presentation. Apart from the contribution to fundamental research, can you imagine any biotech or other applications using these motor complexes?"
Mm-hmm.
And he's thinking of the use of nanopores for sequencing, for example.
Yeah, that's a good one. Yes, you can imagine a lot. Yes, but yeah, if there's more ideas, feel free to.
Okay.
Great.
All right, then we close the poll for the people here in the room. If you want to join, we are going to the restaurant now. So please feel free to join if you want.
Great.
Thank you all for joining.